Well, there's been a lot of important news concerning some viral disease outbreaks, and I thought I'd pick the brain of a great virologist. Joining me now is Vincent Racaniello, Ph.D. Dr. Racaniello is a professor of microbiology and immunology at Columbia University, and he runs the fantastic website Virology blog at virology.ws and a whole slew of podcasts. He's got a media empire. Dr. Racaniello, welcome back to the show, sir.
Thank you, Robert. Happy New Year.
Same to you. It's been a few months, and I want to go ahead and dig right into what's going on with your Zika lab. I went to Zika Diaries, and there hasn't been any updates posted in a couple months. And you had the unsuccessful attempt to secure funding for a study you wanted to do on Zika neurovirulence. So what's been going on in the Racaniello lab in the past couple months?
Well, I stopped writing Zika Diaries over the holidays. I figured people needed a break. And then my virology course here at Columbia started up in January, so I was a little busy with that. And in fact, it was just this week now I put a new post up there on the Zika Diaries all about publishing, because we're writing up a paper, and I thought it would be interesting to discuss with people how we decide where we send the paper to. So I'm back to writing, and you should see more recent, more regular Zika entries from now on.
Okay, great. So you had this one grant, I don't know, what's the right word? Unapproved, disapproved?
Yeah, disapproved. We didn't get our money, basically.
Yeah.
So we resubmitted that because, the criticisms were, we thought, not really valid. So it's back in there, and we'll hear in a couple of months. But in the meantime, we have to write some other grant applications. We're doing one on enterovirus 68, which is another virus that we've been interested in.
Okay.
It's going to be a tough year, but we keep trying. We don't give up.
Yeah. It was really interesting to read one of your last posts and what your comments to them were and what you were actually thinking. I thought that was fantastic.
Well, the blog gives you an opportunity to say things that you might not otherwise say.
Right.
I'm taking advantage of it, especially since I'm kind of near the end of my research career, and it doesn't really matter what I say anymore.
Okay. All right. Well, good. Now, there's been, just because there's not much coming out of the Racaniello lab, there has been a lot of research on Zika. And I was looking at some numbers, and it looks like there's been about 1,400 publications on Zika, and over 1,000 of those have happened just in the past 12 months. So the research is blossoming. There's tons of it. And I wanted to get your thoughts on some of the latest. There was a CDC study about mid-December, and it showed that Zika virus RNA replicated and stayed in the brain tissue of infants with microcephaly and placentas of women who suffered pregnancy loss for at least 7 months after the mothers contracted the virus. Can you explain what they were doing in this study and what your thoughts on it are?
So this is a study out of the CDC where they looked at a number of cases imported into the U.S. from other countries. And these are cases where some of the mothers had confirmed Zika infection during pregnancy, and some of them gave birth to babies with microcephaly. Some of them, the children died. And they took specimens from the placentas and the fetuses to see if there's Zika virus RNA in them by a polymerase chain reaction. And overall, what they found is that in a number of cases, Zika virus is present in children born with microcephaly, so it provides more support for the role of the virus in causing that problem. Interestingly, when the mothers get infected early in pregnancy, the first trimester, that's when you get the most birth defects in the children born. Second and third trimester infections, much less likelihood of a problem. And so that's really interesting. We don't know why that is, but it just tells us that first trimester is an important one for the virus getting in there and doing damage. Now, when the mother's infected in the first trimester, the disease in the mother is transient. It's a rash disease that comes and goes within a week or two. But apparently, in a number of these children who were born, you get the placenta at birth, they can still find viral RNA in there. Yeah. So the virus is sticking around for a long time, Whether it's a problem to the child or not, in later times we don't know. So the virus is unusual that it has a tendency to persist. Now, they also find in children born, some of these children are born and they die, they find virus in the brain. So it could be that if your mother's infected in the first trimester, baby's born, maybe even if you don't see microcephaly, the virus is still there and it could be causing later problems. Mm-hmm. So, this is one of these interim studies. We need to do a lot more of these as children are born to infected mothers to figure out exactly what's going on.
Yeah, one thing that's clear to me as we go through some of these studies is this is just the tip of the iceberg. I mean,
yeah, for sure.
Yeah, no doubt about that. Now, in a related topic, there were a couple of reports that showed that microcephaly and other brain abnormalities in fetuses and infants exposed to the Zika virus during pregnancy occurred more frequently than previously estimated. However, the proportion of the infants with birth defects differed between the two studies. Can you give us a summary of that? Because you can explain this stuff really, really well.
So this is another study of cases where they have confirmed infections in pregnancy. And when they look at, the 3 trimesters, infections in the first trimester, 11% of those lead to birth defects. And these include things that you can see like microcephaly and other major changes to the fetus. So, and again, second and third trimester, not much of a problem. But one of the issues in this study that's brought up is that there are probably more subtle defects in these children who are born to infected mothers than microcephaly. Just because you see a baby born, you say, there's no microcephaly, doesn't mean that the virus has already done some subtle damage that's going to affect this child in its early years of life. Things like cognitive issues aren't going to be seen later. So we're really going to have to do long-term follow-up on these infants born to infected mothers.
Now, of course, other than vaccines, the other issue that's important is getting good diagnostic testing. And
right.
We see that Wadsworth up there in New York and the University of Texas medical branch developed a new test that's more rapid, more accurate. This is some exciting news.
Yeah, so right now the best way to diagnose Zika is by PCR, but you only have about a 2-week window while the virus is replicating. If you want to look longer, you have to look for antibodies. And the problem so far is that the antibodies against Zika cross-react with dengue and West Nile and yellow fever, so there hasn't been a specific test. And what they did at the Wadsworth was to come up with a test detecting antibodies specifically to Zika virus, and that's going to be really good. Now remember, it has to be FDA approved, but as soon as that's done, this will really help diagnose infections.
Yeah, as soon as it's FDA approved, that's when it'll get out to all the hospitals and the Quest and LabCorp and all that. Then there was a study from the Jala Institute for Allergy and Immunology, and it's basically talking about the positive effect of the CD8 plus T cells on controlling Zika infection. What are your thoughts on that?
So here's a paper done in mice. Mice are great models for Zika infection. And they show that CD8 T cells, these are so-called cytotoxic T cells, they actually are important for protecting against Zika virus infection. Most our vaccines work by making antibodies, and they protect you. But it looks like in these mice, it's the CD8 T cells. And they also identify what parts of the virus are recognized by these cells. And why this is important is probably because any vaccine in humans that's going to be effective probably needs to induce a CD8 T cell response, and that's important information.
Good. That's pretty much what I got out of it. Now, I know you're reading research all the time. Is there anything that caught your eye concerning new Zika research?
One paper I really liked was published this month in Cell Reports, and it clarifies a lot of confusion about what the receptor is for Zika virus to get. And as you know, cell receptors are needed for viruses to get into cells. And many papers have said, well, the receptor is AXL, AXL. It's a protein on the cell surface. Well, this paper shows in some cells it's AXL, in astrocytes and in glial cells. but not in neural cells. So there are probably more than one receptor for Zika virus to get into cells. And the cool thing about this report is the virus doesn't actually bind directly to AXL, but it binds to what's the normal ligand for AXL, which is a protein called Gas6. So the virus binds Gas6, and Gas6 binds AXL, and the whole thing gets taken up into the cell. I thought that was a really great study.
Yeah, I saw that also. I'm talking to Dr. Vincent Racaniello with Columbia University, and we're looking at some viral diseases, some viral news, and some viral outbreaks that are going on around the planet. And one of the bigger stories that's been going on during the course of January, leading into February, is yellow fever. And yellow fever is another virus that's transmitted by the Aedes mosquito, the same one that transmits dengue, Zika, chikungunya. Brazil is getting a pretty serious outbreak right now. Some reports say it's the worst outbreak in about 17 years. Dr. Racaniello, can you give us a little primer on the yellow fever virus and yellow fever disease? It's more severe than many of the other mosquito-borne diseases.
Yeah, that's right. Even though it's in the same family as Zika and West Nile virus, it can cause more severe disease because the virus can infect the heart, the liver, and the kidney and destroy those organs. And so you can have a lot of fatality, which you don't normally see in adults infected, say, with Zika virus. So there's lots of yellow fever left globally. WHO estimates about 200,000 cases a year and 30,000 deaths. And so that's why it's a big problem. Vaccines in limited supply. And the issue here is that there's a monkey reservoir for the virus, and that's how this outbreak in Brazil has been occurring. Monkeys get close to people, and the virus is transmitted from the monkey to people, and they get sick.
Yeah. Now, yellow fever was once a big problem in the U.S. I don't think it's been seen since the turn of the 20th century, 1900 or so. Mm-hmm. We have the mosquito vector. We have plenty of travelers to the U.S. From Brazil, for example. And I would suspect a susceptible population, a naive population. Any concern about yellow fever popping up here in the United States?
I don't think so because, Aedes aegypti, also the vector for Zika virus, we've only seen local transmission in Florida and Texas in very few cases. So at the most, I would say that's what could happen with the yellow fever virus. It's not likely to go further north than that.
Right, but we can't say the risk is zero.
Not zero, but certainly pretty low. And of course, we do have a vaccine here in the U.S. I'm sure if there were a small outbreak in Florida or Texas, they'd deploy that right away.
Sure. Now, in addition to the Brazil yellow fever outbreak, one of the bigger viral disease news stories in recent days and weeks is the Seoul virus. And we saw about 8 cases in Illinois and Wisconsin. Again, Dr. Racaniello, can you give a primer on this virus? And I would say this is something that most Americans never heard of.
I think most Americans have heard of C. nombre virus, which is a hantavirus that emerged in the Four Corners region of the U.S. A number of years ago. That's a virus that's transmitted from deer mice to people. You can inhale, or aerosolized urine or feces or saliva. It causes hantavirus pulmonary syndrome. Seoul virus is from the same family, except it doesn't come from deer mice. It comes from rats. And again, the rats are infected. They don't get sick, but they can spread the virus to people. And what's happened, and for this virus in people, it causes not hantavirus pulmonary syndrome, but hemorrhagic fever with a kidney syndrome. And what happens is, in this outbreak in the U.S., 13 states have reported infections, 10 in Illinois and Wisconsin. Pet rats are transmitting the virus to people. There's some rat breeders who have been raising rats and selling them, and unknown to them, the rats are infected because the rats don't get sick. You can't really tell. So this is the source of this infection. And it's going to continue because I think people apparently like to have rats as pets.
Yeah, apparently. Yeah, I didn't even realize there was such a thing as ratteries. I'm like, what on earth is that?
Yeah, it's apparently so. And as you can't tell they're infected, so it's a threat.
But you would agree that the Seoul virus is less pathogenic compared to the sin nombre virus?
Seems to cause less of a serious disease.
Right.
There are fewer cases. But I would say to people, avoid rats.
Yeah. Well, and I guess that leads to my next question, this little tidbit, and I want to see if you had any concern about this. The CDC says that this is the first known outbreak associated with pet rats in the U.S.
That's right.
Any concern about that?
Well, yeah, I think that, we're always seeing new viruses popping up like this. The question is, have they been around before and we just didn't recognize them? I think people have always probably had pet rats, but maybe we didn't recognize the syndrome before, or maybe this is really something new. We'll see. I do hope that we can use ways of diagnosing the rats to make sure that, these ratteries are virus-free.
Okay. Now, I'm not sure if you're aware of this, but there's been some measles up in your part of the woods.
I did see that.
Yeah. So I saw one in Long Island and a couple cases in northern Jersey.
Right.
I've talked about measles on here 100 times. So, as a virologist with decades of experience, do you believe there's a more communicable virus than measles, or is that the top of the heap?
It's pretty high up there. I always tell my medical student classes, I'm standing in front of 150 students, and I said, if I had measles, you'd all be infected by the end of the lecture. And that's because it's a respiratory transmitted virus, but it's just really good at getting from person to person. And if there's some unimmunized people, they're going to pick it up, and that seems to be what happened here.
Now, is there any other virus that compares to that?
I don't think many come close. I think smallpox is pretty transmissible as well, but I don't think it comes close to measles. Flu is way below these. Flu doesn't, even though flu gets a lot of news, a lot of numbers of cases, it's not as transmissible as measles.
Yeah. Okay, well, here's I got about, till the break, I got about a minute and a half.
Okay.
And January marks 6 years since the last polio case reported in India, and this is near and dear to your heart. In about a minute, can you talk about the significance of this milestone?
So as you know, WHO wants to eradicate polio. They've been working on it since 1988. And India has been a tough nut to crack for years and years. There were huge efforts to vaccinate in India, never made much of a difference. Kids were getting 5, 8, 10 doses of vaccine. And there was some thought that we would never be able to get rid of the virus, which transmitted freely. Yet, as you say, 6 years ago, they did it. It's an amazing accomplishment. The significance is, if we can do it in India, we can do it anywhere. And the only countries left now where there's still wild polio are Afghanistan, Pakistan, and Nigeria. And the only reason the virus isn't gone from them is we have armed conflict that prevents the vaccinators from getting in. If we could get in there, India proves that we could immunize and get rid of polio.
Yeah, it's an incredible story. I remember first reporting on it when it first happened, and you kind of like sit back and wait. Well, how long is this going to last? Well, it's lasted so far.
I think it's going to happen. It's just a matter of time, and that's going to be some outcome, eradicating another viral disease.
Okay. Oh, so you time frame, when will polio be eradicated?
Well, I think it's going to take another 5 or 10 years because this conflict isn't easy to fix.
Right. All right, Dr. Racaniello, can you hold on till after the break? I got a few more questions.
You bet.
All right. Again, I want you to check out the website, outbreaknewstoday.com, outbreaknewstoday.com, and I encourage you to check out Dr. Racaniello's website. It's virology.ws, and the title of the website is Virology Blog. Great resource on all things virology. And I will see you after the break with more with Dr. Vincent Racaniello.
Welcome back to Outbreak News This Week, your source for all the news about worms and germs. Here's your host, microbiologist and editor of OutbreakNewsToday.com, Robert Harriman.
Well, welcome back to the show. I am being joined today by virologist, writer, media guru, and professor, Dr. Vincent Racaniello from Columbia University. And we've been talking about some virus news, some virus outbreak information. And I want to continue our discussion. And there was a very interesting study that came out very recently concerning why some individuals get the more severe and sometimes life-threatening dengue hemorrhagic fever. Vincent, you're familiar with that study, correct?
I am, yeah.
Yeah. Can you talk about that for a second?
So in dengue, you get an infection. There are 4 serotypes. You typically recover. You make antibodies to whatever serotype infected you. Then if you get infected with a different serotype, then you make antibodies to the first one. They bind the second virus, but they don't prevent it from infecting. In fact, they make the disease worse, and that's the severe dengue that we're trying to prevent. In this study, they looked in people with dengue secondary infections, and they looked at their antibodies, and they found that some patients that have severe disease have a very unusual antibody. It's a variant antibody that lacks a sugar in a part of the molecule that's known to activate immune cells. And the idea is that without this sugar, the antibodies cause destruction of platelets. That leads to bleeding, and of course bleeding is a hallmark of hemorrhagic fever, which is one of the problems with this severe dengue. So this is a really interesting finding. First time this has been shown, and I think it makes it possible that perhaps we could identify the patients who are at risk for more severe dengue, because it's not everyone, and then we could take care of them differently. So
Sure.
Pretty exciting study.
Yeah, I thought so too. Let's go ahead and take a look at some of the stuff you're doing over at virology.ws, Virology blog. And you're doing a fantastic job. And I actually watched one of your videos recently. I see that you're doing it for the ASM, the American Society for Microbiology now.
Yeah, so we have, from time to time, I go to meetings and interview people, and they send a camera crew, so we do some interviews with people who are doing interesting virology out there. Oh,
congratulations. That's great. And I really enjoyed the interview with Dr. Jana Mazet. I hope I said that right. Mazet, yeah. And she's does a lot of research on emerging infectious diseases. And can you give my audience a summary of your discussion with Dr. Mazet?
Sure. So Dr. Mazet is running a global project. It's called the Novel Viral Emergence Early Warning Project, or PREDICT. And what they're trying to do is go to places where viruses typically spill over from animals into people and collect lots of specimens and find out what viruses are out there. She wants to know all the potentially dangerous viruses all over the planet. And so far, in a couple of studies that they've done, she said she's discovered 800 new viruses that weren't known before. Now, what she'd like to do is kind of, if you remember the Human Genome Project, which was done over 10 years ago, it was proposed for $3 billion. We were going to sequence that first genome. Well, she wants to do the same thing for the virome, and she figures it'll cost about $3.5 billion. She wants to spread the costs across the world, but most importantly, she wants to put technology into countries so that the work can be done there. She doesn't want us just to grab samples and leave. She wants us to enable countries to do their own sequencing and PCR. So, it's a tough sell to tell people we want to spend all this money to find all the viruses out there, but, I think it's a great project.
Yeah. And I don't know if maybe if people were doing this before, would we had a better heads up on, say, Zika?
I think we need to know what's out there.
Yeah.
Right? If you don't, you can't do anything. So, we sort of knew about Zika. We didn't know how it was spreading as it was. But there are certainly other viruses out there that we should point at and say, hey, this one has potential. So,
sure. But even with Zika, the amount of information, about the amount of research that was done prior to 2015 was negligible.
It's unbelievable how it exploded.
Yeah. I also noticed on your website, Virology blog, that you're doing more writing about prions.
We are. Prions and bacteria. In fact, today I just wrote a new post. They found prions in bacteria, of all things. Prions in humans cause transmissible neurological diseases, right, like Kuru and Creutzfeldt-Jakob. But it turns out that there are lots of prions out there that don't cause disease, and they just found them in bacteria, so that's really interesting.
Yeah.
I also wrote about a blood test that's been developed for detecting prions. The problem is we can't detect the disease until it's symptomatic, and then it's too late. But if we could find the prions in the blood, we could be ready for it. And there were a couple of assays that have been developed that look good in terms of that.
Yeah, and actually you allowed me to republish that story on my website, and I appreciated that. That was very good. So what else exciting and new are you doing on Virology blog? It looks like in the podcast it looks like you're doing a lot of podcasts now.
Yeah, so at the blog, I write a post a week, but one of the main activities is podcasting, and I have a little media empire, like you call it, where we do This Week in Virology, This Week in Microbiology, This Week in Parasitism, This Week in Evolution, and a couple of others. And it's all about teaching people about viruses, bacteria, and so forth in a way that they can understand. I've even started a short video series, 8-minute videos trying to explain viruses. So they all come out at virology.ws. I also am teaching my course in virology this semester at Columbia University, and I record all my lectures, and they go onto YouTube and Virology blog as well. So if you want to learn about viruses, you should check those out.
Yeah, good stuff. It's funny. During the day while I was at the lab working, I was listening to one of your, I guess you call it TWIM, This Week in Microbiology.
That's right.
Off of Stitcher. And the topic was about one of your guests was talking about bleach and cleaning laundry and Germany, and it was the most fascinating topic. And I was like, oh my.
Well, laundry practices are changing. We use lower temperatures and less detergent. So the question is that having an effect on the microbes on our clothes? And that was what that discussion was all about.
Yeah, fascinating stuff. All right. Well, thank you, Dr. Vincent Racaniello, for your time and expertise and all the interesting stuff you laid on us today. I appreciate it very much.
Robert, always a pleasure, and, the microbes are just fascinating.
Yes, sir. All right. I appreciate it so much. Thank you, Vincent.
You're welcome.
All right.